Dear students, in our series of basic courses, we will continue the course of immunology. And today we will discuss the function of cytotoxic T cells. So first of all, we will see what we will be able to learn after this lecture. So we will see which type of cells are killed by the cytotoxic T cells, why they are called or why they are named as the cytotoxic T cells, and at the end we will see how cytotoxic T cell perform their killing functions. So if you look for here, after explaining the previous lecture, as we know that the T cells of the two types, helper T cells and the cytotoxic T cells. So in our last lecture, what we have learned, we have discussed the functions and even the differentiation of helper T cells. So before starting our next lecture, so let's recall once what we have learned from the last lecture. So we have studied actually the four major subtypes of helper T cells, such as Th1, Th2, Th17, and T regulatory cells. And then we also discussed how they got differentiated. So they get differentiated with the help of different kind of cytokines, such as Th1 by the interleukin 12, Th2 by interleukin 4, while Th7 by interleukin 6 and TGF beta. And we also discussed in our previous lecture what are their major or main transcriptional regulator. So we know that, and we have already learned that in case of the Th1, we mentioned that they activate the state 4. In case of the Th2, there was a GOTA3. In case of the Th17, we discussed about the state 3, and T regulatory cells, their master transcription regulator was FOXP3. So, and then we also discuss when these cytokines differentiate the helper T cells into different subsets, and then they activate their master transcriptional regulators. And as a result of that one, then they induce or maybe involved in the production of certain kind of cytokines. So such as the Th1 involved in the production of IFN gamma and interleukin 2. While in case of Th2, it secreted the cytokines such as IL-4, IL-5, and IL-13. But if we talk about the Th17, so Th17 involved in the secretion of IL-17 and IL-12. And then last one, which was the T regulatory cells, so they secrete the IL-10 and TGF beta. So what they perform the major function. So in case of Th1, they were involved in activate the macrophage. In the case of the Th2, they activate the mouse cells and eosinophils, and then they of course they kill the infected cells. And while the Th17, they produce the antimicrobial peptides. While in case of the T regulatory cells, they are involved in the suppression of immune systems because they start working when the pathogen was already eradicated. So that was the whole story which we have already learned in our previous lecture. So here the purpose was because that lecture was totally long, also slightly complex. Therefore, I provided for the summary of the last lecture. So now we can proceed our today's lecture. So today's lecture is slightly simple as compared to our last lecture because the last lecture we have the helper T cells got differentiated in the various subsets, but in case of the T cells, the story is slightly simple as compared to the helper T cells. So if we talk about here, what we should recall even before starting our today's lecture. So one thing we have learned in our previous lectures, and we know that the CD8 positive T cell form a cytotoxic T cells. What they do, which directly kill the infected cells. So when we have know that, so in this lecture, what we will try to understand the cytotoxic T cells. So what we should recall before proceeding. So we have already discussed what we have recalled. We mentioned that activates CD8 positive T cells form a cytotoxic T cells. And after the understanding of activated CD8 positive T cells, what we have discussed, what they do, is they form a cytotoxic T cells. So on that, there are the two questions that come to our mind. So first, why they are called a cytotoxic T cells, and other, what are the function of these cytotoxic T cells. So today's lecture will give answer to these questions. So if you look for that, why they are known as CTLs and why they are called the cytotoxic T cells, and even they are also called the killer T cells. So we have given this name? The name was given because they directly kill the cells that display the foreign antigen and directly kill the virus infected cells or tumor cells. What can the tumor cells? That display mutated peptide on their surfaces. So that's why what we call them, that's why we are call them as a cytotoxic T cells. So how actually the story was started, if we strongly goes back to our previous lectures, so we know that when there is an intracellular infection, so what happened? That is recognized by CD4 T cells, and then these CD4 T cells got differentiated into a special kind of subsets that is called Th1 subsets, and then actually that Th1 subset release a special kind of cytokines that is called interleukin 2, and then that interleukin 2 actually send the message to the CD8 positive T cells, and then as a result of that, CD8 positive T cells got differentiated into a toxic T cells, or what we call them as a cytotoxic T cells, and they're also called because they are going to kill the pathogens, so they are actually known as a effector T cells. So under the normal conditions, if they have a self-antigen, so cytotoxic T cell doesn't perform their function. But when there is an infections and there is a foreign antigens, then these cytotoxic T cell perform their functions. And when they perform their function, those cells, because they are directly targeting that cells, so that's why those cells which are targeted by the T cells or killer T cells, we give them as a target cells. So what they do, it means activated killer T cells, they directly in contact with the target cell, and then they kill those targeted cells. And as a result, the target cell were destroyed. So now, of course, the question is that okay, how uh, uh, these cells was activated, and then as more importantly, what we are going to talk in our today's lecture, okay, how when they come to tract, how the killer T cells kill those infected cells and directly. So if we talk about that, how cytotoxic T cells work to kill those infected cells. So after the activation and clonal expansion, which we have discussed in our previous lectures, so these T cells form a two kind of cells. One we are called as effector T cells, and other we called as a memory T cells. So because these cells are directly involving in the killing of cells, it means these are the effector T cells. And now what happening actually after the clonal expansion, these effector cytotoxic T cell leave the lymph nodes and they start to migrate toward the site of infection. And at the site of infection, what they does is to perform its killing function. It means the cytotoxic T cells direct contact with the infected cells. As you can see that, let's suppose this is an infected cells and this cells have a target cell displaying the foreign antigens. But in this case, if you remembered our previous lectures, we know that the cytotoxic T cells or even CD8 positive cells, the antigen was provided by the MSC class 1 molecules. So what happened in this case? These cytotoxic T cells get bind to the foreign antigens via TCR. So what is a TCR? We know that the T cell receptor. So simplify, we called it as a TCR. As you can see here, that structures we already know that we call it as a TCR or T cell receptors. That is on the surface of infected cells. And then what happening against? In addition to that, there is also CD8 co-receptors that bind to the MSC class 1 molecules. So now the cytotoxic T cells undergo another process that is called the degranulation. So what happening here, what we have learned so far, or in mean the terms of what we are going to answer, we are going to answer the questions how cytotoxic T cells, or simply we call it the CTL, work to kill the infected cells. So what we have talked so far, we mentioned that when the infected cell, what kind of infected cells? The target cell displaying the foreign antigens to make the direct contact with the infected cells via TCR or co-receptor, that is in case is a CD8. And upon the interactions, CTL, it means the cytotoxic T cells undergo the degranulation, and that granular contents and that contain the perforins, granzymes, and granulogens, and these three kinds of granular contents are released. So now we will try to understand what actually they does. So if you look for here, these are the cytotoxic T lymphocytes, and they release, they go under the process of degranulation, and as a result, they release the perforins. And so, what does the function performed by these perforins? Actually, if you look for here, these perforins, as their name is indicated, that they form a pores in the membrane. And what happening as a result of these pores or via these pores, the other granular contents could enter inside that infected cells, such as granzymes and granulogens could enter inside that infected cells, and then they induce the apoptosis, that is also called what we simply call it programmed cell death. So, so in the simplify, what we say that and what we have learned here is cytotoxic T cell bind to the foreign antigen. How they bind by the TCR, and other is on the surface of infected cell, and CD8 co-receptors that is bind in the, and that is presented in the form of complex, in the which kind of complex, MSC class 1, and that foreign antigen peptide complex. And now what happening after that is cytotoxic T cells undergo the degranulation, releasing the contents of the granules. So what kind of the granules? We mentioned that there are the following granules such as the perforins, granzymes, and granulogens. So what they do actually, perforins release, make the pores inside the membrane, and because of that pores, other two granular contents like a granzymes and granulogens could enter inside that infected cells. And then as a result, what they do, they cause the apoptosis, or that cell undergo the apoptosis. It means natural programmed cell death. And then these proteins are synthesized and loaded into the granules as naive CD8 cells encountered a specific antigens. And these enzymes perform a following functions, which we have already discussed. So we just want to memorize what these granular contents perform the functions. So we already mentioned that a perforin form the pore in the membrane of the target cells, which damming the target cell membrane and form a passage of entry of other enzymes. What kind of other enzymes? Granzymes and granulogens. And now the granzymes enter through these pores and created by the perforins and cause apoptosis via mitochondrial dysfunction. And what the function does by granulogens? So granulogens antimicrobial activity and contribute toward the apoptosis. So these apoptotic bodies are engulfed by the phagocytes, and while the cytotoxic T cells move toward, as you can see that, we have a macro, here we have the cells, they start to engulf the cells by the phagocytosis process, and these cytotoxic T cells move to the other cells to perform their functions. So after killing that cell, so they go to another infected cells and they start working there. Before I want to mention here one point, that in addition to this function, these cytotoxic T cells also release some kind of cytokines. So as we mentioned that in case of the helper T cells, they are released, every subset of the helper T cells release a various kind of the cytokines. But in case of the cytokine T lymphocytes, so what kind of the cytokine released by the cytotoxic T lymphocytes? So what they release, they actually release the tumor necrosis factor alpha and IFN gamma. And what they does actually, so these cytokines activate the macrophages and recruit them to the site of infections. So this is all the story for the what we call T lymphocytes. Let's summarize our whole today's lecture. So what we have learned so far. So firstly, we know that a cytotoxic T cell directly kill the viruses infected cells and or tumor cells. And then how they perform their function? So to perform their function, cytotoxic T cell directly contact the target cells and they release the cytotoxic contents. And what kind of the cytotoxic contents? By the process of degranulation. And these are the perforins, granzymes, and granulolyzins. And we also studies what actually these contents does. So perforins from the pore in the membrane of the target cells. Why? What the granzyme does? So it enters through those pores created by the perforin and causes the apoptosis via mitochondrial dysfunctions. And lastly, the granulations, what it does, it induces antimicrobial activity and then contribute toward the apoptosis. So along with this, so we have learned that how the cytotoxic T cell perform their function. So this is the whole story. So we discussed the both types of the T cells, helper T cells in our last lecture, and in today's lecture we discuss about the cytotoxic T cells. So we will see what we are going to learn in the next lecture. In so far we have the understanding how when the pathogen enters our immune system responds. So and then the story was started with the transplantation rejection. So now again we come that if the but now we know that transplantation is possible. We can transplant the several organs. So how that transplantation has been made successful. So that's we are going to learn in our next lecture. In other terms, we can say that what we are going to learn in our next lecture, that is a transplantation immunology.